This video offers a visual guide to key bioinformatics topics, including DNA barcoding, metagenomics, whole genome bisulfite sequencing, WGBS, and eValue. These tools are crucial for species identification, understanding microbial communities, studying DNA methylation, and assessing sequence alignment significance. We'll explore the production pipelines and applications of these methods, covering topics from DNA barcoding for rapid species identification to SNP detection and gene expression analysis. The summary box highlights how these tools collectively provide crucial biological insights. DNA barcoding is a rapid method for species identification using short DNA sequences, often one or a few gene regions. The production pipeline involves sample collection, sequence amplification, barcode generation, and comparison with online databases. Standard barcode regions include CO1 for animals and RBCL, MATK, TRN HPSBA, and ITS for plants. Key steps in the production pipeline ensure accurate data for comparison and species identification. The process begins with sample collection and correct labeling. Sequence amplification, generating many copies of the target DNA region, is followed by barcode generation and final comparison with reference databases to identify the species. Good barcode regions must show species-level variation, have conserved flanking sites for universal PCR primers, and be short in length. Accurate data depends on quality samples and labeling. DNA barcoding is a fast and reliable tool for species identification. The production pipeline, from sample collection to submission, ensures accurate and reproducible results, supporting applications like biodiversity conservation. The four main steps, collection, amplification, generation, and comparison, are universal. Online barcode libraries like BOLD are essential for matching sequences. Remember, a single correct identification can make a worldwide impact. Metagenomics studies the total genetic material of microbial communities directly from environmental samples. WGBS analyzes DNA methylation. eValue assesses the significance of sequence alignment results. Metagenomics approaches include single-gene surveys and random shotgun metagenomics. WGBS involves bisulfite treatment to study DNA methylation. Bioinformatics tools like NCBI and eValue are vital for interpreting data and assessing sequence matches. Metagenomics aims to measure diversity and discover new genes. WGBS reveals epigenetic regulation. eValue helps determine sequence significance. Primer design and SNP analysis are crucial for studying genetic variation. Metagenomics goals include studying diversity and ecological relationships. WGBS is a technique for whole genome DNA methylation analysis. Useful bioinformatics tools include eValue, PrimerBlast, and DBSNP for sequence interpretation. Key bioinformatics topics, DNA barcoding, metagenomics, WGBS, and sequence analysis are interconnected. They provide the tools for identifying species, analyzing microbial communities, studying epigenetics, and assessing genetic variation. DNA barcoding uses short sequences like CO1 in animals and RBCL/MATK/ITS in plants. The process includes sample collection, PCR amplification, barcode generation, comparison, and submission to the BOLD database. These topics link laboratory methods with sequence databases and computational analysis. They support species identification, microbial community study, epigenetic analysis, and gene expression research.